just wondering if we could talk a little bit about ADCs and
Certainly.
your interest
Sure.
in ADCs.
Sure.
So should we start with why there has been a pickup of interest?
Sure. So I think really the data coming out of the two lead compounds, which would be the Seattle Genetics CGM35 compound and Genentech's TDM1, Roche I should say is TDM1, have been so compelling in terms of both their efficacy profile and safety profile that it's hard to ignore. And so they've really showed eye-opening results in their phase 2 trials and what's been published, I think, limited in their phase 3 trials, and I think the whole field is very much anticipating both of those molecules becoming approved products in the next 1 to 2 years. And so I think with that you'll see the whole field kind of open up in terms of interest, and it already has. There's probably a dozen companies now that have ADC programs using both the Seattle Genetics and the Immunogen technologies.
Okay. And so at Takeda, you're obviously very interested in ADCs.
Yes, we are.
And are you working with ADCs?
We are, yeah. We have several. I mean, as you know, Takeda has rest of world rights for the Seattle Genetics compound, and I think we're looking to leverage off of that in terms of expanding not only our stable of portfolio products using ADCs, but I think looking long term, we look at this as a growth area and potentially highly beneficial for both hematologic malignancies as well as solid tumors.
Okay. So where's your personal interest lie? Do you think that there is more promise developing ADCs for solid tumors or liquid?
Well, I think based on what's been published so far, and it's interesting that the two leaders, one is a solid tumor indication and one's liquid, so I think there's optimism and room for growth in both areas. As you know, naked antibodies have been predominantly successful in liquid tumors and hematologic tumors, and so I think the next kind of holy grail, if you will, in oncology is to have solid tumor indications, and I think the promise is, of course, this ability to deliver highly potent chemotherapeutic agents with the specificity of an antibody, and in many ways that would kind of fulfill the magic bullet promise that antibodies had 30 years ago and never really delivered on.
So could we quantify this? How much more of an improvement could an ADC give over a naked antibody?
I think that's one of the key questions, and obviously that will be a case-by-case basis, I think, by disease, by tumor type. But I think what's really going to be intriguing over the coming years is the durability of response. And so now you've seen, kind of the first wave of you get tremendous responses, and then how long those responses last will be critical, and that's a function of time. So you're going to have to follow these patients over a prolonged period of time. But if they also get, the durability of response, and I think that's tremendously exciting, right? Because in theory, those patients don't require any more additional therapy, so that from a quality of life, public health, cost of care perspective, all of those things could be hugely impacted by this type of therapy.
What do you think the next couple of years look like for ADCs? I know the PDUFA date's coming up.
Yeah, so I think, again, with the two leading compounds that are out there and the ones that have the most public visibility, if in fact they are both approved, I would see the field just absolutely exploding in terms of the number of compounds in there and the number of companies trying to get in on this type of therapeutic area. And I think you'll see expanded indications, you'll see new target antibody combinations coming forward. And I think eventually you'll see an evolution of new drug linker technology as well. And so one of the key components that I see is these two leading candidates now are both mitotic inhibitors through tubulin formation. To me, it would be nice, and I know that everybody's working on it, to have multiple combinations of things, different drug linker combinations that have different mechanisms of action, so DNA interpolation, those types of mechanisms, as well as tubulin inhibition.
And so how have we got to this point where we've got two promising candidates? I know that there must have been lots of technical and engineering issues that had to be overcome.
Yes. And I think obviously, in many ways, in my mind, it's fields that have kind of co-evolved, right? So you've gotten to a point where you have very robust and stable expression systems that can deliver you multi-gram quantities of antibody in a cost-effective way. There's been significant improvement in the drug linker technology, which I think was probably the most significant advancement. And it's getting to the point now where you can actually customize an ADC candidate to have either a cleavable or non-cleavable linker, a variety of different things. And so I think the composite of those things together is really what's gotten us to this point. And I think there's an almost limited number of possibilities, I think, particularly if these first couple of compounds get approved in terms of what will happen both at the basic research level but also in the clinical development and clinical investigation side.
And can I ask you about the role of ADC Summit? Is it a mark that people are seeking out interest?
Yeah, I think, well, it does a couple of things. Number one, it kind of gives you some visibility to what other people are doing. It's a forum for addressing kind of common issues that you're facing either in development, manufacturing, etc. It's a way to get a sense of regulatory approaches that are going to be required, analytical approaches that are going to be required. And so in that way, it's a nice way to kind of gather anybody who's kind of in the field together to kind of exchange information and get an understanding of your approach and how it relates to others for some of those common problems.
Great. Thank you.